The first afternoon session is going to be by Yaël Balbastre. Yaël is currently an instructor in radiology at Massachusetts General Hospital and Harvard Medical School, where he develops computational methods tailored to ex vivo images of the human brain. He received his PhD from Paris-Saclay University in 2016 and was then a postdoctoral researcher at the University College London. Research touches multiple facets of medical imaging, such as segmentation, registration, shape modeling, and quantitative imaging. Over to you.
Hello, everyone. Well, thanks for organizing this workshop. It's super informative. Let me share my screen. Yes. Well, I didn't realize that was just after Urania, so I guess you guessed where I'm going to go with this. I'm going to talk about learning to segment vessels in the absence of manual labels. And I won't talk about vessel that much, first because I actually don't know much about it. You know much more than me. And I wanted to talk a bit more about why we're doing things that way. So I'll probably say a few things in common with Urania, but hopefully in a different way, and that may help. So I'll start with just a very short history of what people have been doing for image segmentation. So image segmentation is really just a mapping between an image of intensities and an image of labels. And so it's a pixel-wise classification problem. So that's in computer vision. In medical imaging, the main difference is we often work with 3D images. So we have voxels instead of pixels. And we want to segment these anatomical areas that have some anatomical meaning. So sometimes we're lucky, and the intensities have a quantitative interpretation, and that's the case, for example, for CT, where the intensities follow the Hounsfield scale. And if I want to segment the bone, I can almost just threshold the image between in the range of intensities that I know matches the bone density and get an okay segmentation. Most of the time it's not the case, and we don't have this quantitative mapping between intensities and semantics. So what a lot of people have been doing in the 90s and early 2000s is to essentially guess from the histogram where to set those thresholds, and that's first from my PhD advisor in the 90s, very complicated ways of trying to find modes in a histogram that are robust to noise. Then there were a bit more robust methods based on statistical theory. So whatever, K-means, Gaussian mixture modeling, but it's still essentially, in those cases, we're still setting linear boundaries in intensity space to segment classes. But I mean, you can guess that in a lot of modalities, there's no such easy mapping, linear mapping between intensity and classes. And so what, essentially, if you're looking at segmentation challenges in MICAI before 2015, what people will be doing is compute a filter bank on an image, so compute a bunch of different filters. So it's essentially lifting this 1D signal into a higher dimensional signal, and then train a pretty simple function, so often run some sort of random forest to predict the label based on those filter responses. And so the idea is really to lift this image in a space where we can find a linear boundary between classes. And then in 2015, there's big change, and like the first thing is that, well, those filters can be learned too. They're just like another component of the function, and we can actually stack those things together, and that's a deep network. And so in 2015, there's the first fully conventional network for semantic segmentation. And what I'm doing is saying, well, I can just take a patch and then classify what I'm taking. I'm taking one of these super exciting classification network, and what I'm going to classify is just the class of the center pixel in that patch. And I did that, and I got pretty good results. The only thing is that in that case, you get the independent classification of each pixel. And so oftentimes they would have to smooth the revolutionistic segmentation using some sort of Markov random field, for example. And then if you actually combine this smoothing of the segmentation with this convolutional encoder, you get a unit. And I mean, since then, there's been a few tweaks, but it's basically still the workhorse of semantic segmentation in medical imaging. There's like we're almost frozen in 2015 in terms of the architecture of things that work. And if I simplify now, the way all of these methods work is really we have a function parameterized by some weights, and we're just finding the weights that minimize the distance between the prediction and the true labels on some training data set. So that's one family of methods for image segmentation. And the other family, which Eugenio also mentioned, is based on Bayesian statistics. And that's where I also come from. So I like to think of things in this domain. So we go back to our paired images, right? Images and labels that I call X and Z. And if you're Bayesian, all you need is this quantity, the joint density of images and labels. And if you know that thing, and you only have an image and want to find a segmentation, you can just compute the posterior distribution and spice rule. And it's something that you compute from the joint density. So it's really just, instead of being some prediction, it just becomes calculus. So often, until now, I didn't have, like, this density doesn't have a form. Often, to give it an analytical form, we introduce another variable that we can marginalize, that's this sum here. And we can, again, extend this using the chain rule. It becomes a bit ugly to write, and so we just use this representation, which is a directed acyclical graph. And so all this, what this says is just that we have this joint distribution on image labels and something that I call traits, which is just like something else that we don't really care about, but has an impact on the image and the label. And see, from about 2000 to 2015, that was what all of the methods for brain segmentation that actually worked were using. So it's what's used in FreeSurfer, what's used in SPM, what's used in FSL. And in that case, we have the image, the label, and the traits are essentially a prior map of where to find different classes in the field of view, and then a Gaussian mixture model that maps labels to intensities. And that works quite well, and was like nobody really questioned it for 15 years. The surprising thing is that if you actually sample from this model, so if you fix your means, like your Gaussians and your prior, and you just sample from the model, you don't really get realistic images. That's what you get on the right, which doesn't really look like anything on the left. But even if your joint density is not very accurate, using that density to invert the model and get the labels actually works very well. And then you could ask maybe why we care about those posterior distributions. I mean, that's not really what we want. What we care about is the actual label that we assign to the voxel. But the thing that if you minimize risk, so this R01 function is the risk of making an error when we segment, so essentially a measure of accuracy. If you want to maximize accuracy or minimize this R01 risk, the function that minimizes this thing is just the maximum probability classification under the posterior. So we know that the posterior is actually really good at thing, and you compare it to what we wrote first about the other family of methods, they really look the same, right? We have this minimization between some parameterized function of X and our target labels. And so you can guess that if our parameterized function is flexible enough, what we're going to get is actually something that looks like the posterior distribution. So if we compare these two families of methods, the generative one, we define the joint posterior, and then we define the joint distribution, and we compute the posterior from it. In discriminative methods, we defined a approximate posterior using a family of functions, and then we just compute the weights that make it look as close as possible from the true posterior. And Another thing that's important when we implement our algorithms is invariance and equivariance. And I'll just try to define it very quickly. So invariance means that if we have a function that in this case counts how many sides we have in a shape, if we change the color, we don't want the answer to change. So we want this function to be invariant to color change. If we translate that to medical imaging, it means that if we have different contrasts or different resolution or different artifacts, we still want our segmentation network to give us the same answer up to uncertainty. Equivariance means that is related to functions that actually have to be applied to the output as well. So if I have this sort of edge filter, if I rotate my input shape, I also want to rotate my output. And again, if we translate that to medical imaging, it means that if we had different head position, for example, we want the same segmentation but rotated. If we have difference in anatomies, we want the segmentation to reflect the difference in anatomy. It's actually pretty hard to build this type of invariance and equivariance inside algorithms. So if we use generative models, so Bayesian methods, the way to enforce that is to have what we call a flat prior on some of the traits. So in our case, if we say that that our Gaussian parameters can take any value, it means that this theta gets essentially fed to the image and the segmentation is invariant to it. It corresponds to what Yaël was showing earlier. So the issues with generative models is only very simple models have a procedure that can actually be computed. So we have to rely to approximate methods where we don't compute the true procedure, but we just try to find a function that looks like it. Or we can use a sampling method, so Monte Carlo methods, but are very, very slow, especially if we're working with high dimensional data, such as images. And both of them require this joint distribution to have an analytical form. We need some way to write it mathematically. The alternative is to use a discriminative model, so one of these flexible functions that we feed to the data. But often, so we actually don't have the distribution itself. We just have a big data set of images that we can sample from, right? So I can ask for one pair of image and label, then the next one, and another one, and another one. The issue is that these pair of images actually depend on some characteristics, but we don't have a control on this characteristic, right? So if I only have T1 weighted images in my data set, then I can't control contrast. Now only showing one contrast to the algorithm, to the network. So that's a good thing. So we can use this family of losses that ensure that what we get is something that looks like the posterior. We actually don't need to be able to write the distribution analytically. We just need a big data set that we can sample from. It's flexible, but in practice, we never have an infinite distribution. We only have a few examples. And we can't control traits, so we can't build invariance to it. What we would like is to get back control over traits and be able to essentially say, oh, I want a flat prior of a contrast and be able to sample from my dataset with any contrast. And so what we do is actually go back to our generative model and use that to sample. But instead of using it to invert the probability and get the posterior, we just use it to sample and give examples to the network. So we still have the same good things, still no analytical distribution, but we can build in invariance and equivariance. And that's what Yaël did, and he showed that in depth just before. So we can use it to segment for segmentation. But Malte Hoffman in our lab also showed that we can use it for registration. So we can show example of brains, make fake brains, learn to register them. But so we actually don't even need to show proper anatomy. Like if we're just training a network using those fake shapes, and then apply it to proper organs, it does a good job because the network learns that is invariant to anatomy essentially. And so to summarize, the point of using this synthetic data is that this is the true distribution of images that we want to process at test time. And it's got some shape. Often when we train on real data, we only have access to a a subpart of this space. So there's a bunch of images that are never shown during training, and then that creates a domain gap. When we use synthetic data, we actually try to create a distribution that is much broader than the true distribution, so that when it sees real data, it's inside the space of things that are sampled. But what we're sampling out doesn't need to be realistic. It just needs to be more broad than the true distribution of data. Okay, and so now I'm going to try to explain how we use that for segment vessels. And the reason why we got interested into vessel segmentation is we have a bunch of projects like this one, which is going to be presented by Bruce tomorrow, where we acquired data with different types of modalities post-mortem on a human brain. So usually we do these whole hemisphere MRI with quite high resolution, and then we block out, so we cut a piece of tissue on which we acquire different types of microscopy. So one is OCT, which is quite known for retinal, to image a retina, but we actually use it on pieces of brain, and then light sheet fluorescence microscopy. But the issue is that at each of these steps, there are a bunch of deformations that arise. And so if we want to put everything back into the same space, we need to correct for these deformations. If you look at the different images, like the contrast mechanism is very different. So we can't really use intensity to register these blocks. So what we use is stuff that we can actually see in all of the modalities, and they are vessels. And so this is an MRI, an OCT, a light sheet microscopy, images of the same tissue. And you can see that some vessels are actually present in all of the modalities. And so if we can segment them, then we can use that to drive the registration between the different modalities. And even in the MRI, we actually see, even if it's about 100 micron resolution, we can see quite a lot of vessels, although there can be sometimes pretty close to the noise floor. So we have these different types of modalities in which we want to segment vessels. The issue we had was that there are scales at which there's not a lot of stuff available, because typically people would say, for the brain, people would segment vessels in time of flight angiography, so I don't know, like 1.5 millimeter maybe, or 1 millimeter, or they would segment vessels in microscopy acquired in mice. Not a lot is done in humans, and so we have all of these different scales between 1 millimeter and 1 micron, but there's no real methods available to segment them, and also not a lot of label data to train algorithms. So what we could do is try to synthesize realistic vasculature. There's a lot of methods that exist. This one is from Gonzalo's paper. That's called constructive optimization. And the idea is to essentially grow vessels and optimize synthetic trees under some function that optimize both statistical features, like statistics about how vessels branch out, and also some function of how the physiology works. And so that creates very realistic networks, but we didn't use that for 2 reasons. First, we didn't know anything about them, how to make them, but also there's the risk that these realistic vasculature are more peaked than the true distribution of things that we can see in our data. So we wanted to go back to something that is very broad, maybe not realistic, but encompasses features that are sufficient to segment vessels. And if you go back to what you all used to do, what's used a lot for vessel segmentation are these Frenchy filters or Vesselness filters. And the idea is that vessels are the only things in the brain that are both thin and long. And so if we compute very localized filters, we can actually lift the image into a space in which, when there's not too much noise, vessel can be segmented. And so we wanted to train something that is as flexible as a Frenchy filter, but has a bit more context. So we're going to train a UNet, but we're going to, instead of just computing filters, we're going to just show a bunch of shapes that look like vessels. So to make them, we don't try to optimize anything that makes them very realistic. We just start with endpoints, add a bit of tortuosity, change the radius, and then branch out and continue. And so that's what we get in the end. That's what we train on. We can play with hyperparameters, so the density of the splines. We can play with the radius. We can have something a bit more hierarchical, where we have thinner and thinner vessels that branch out. We can train using the center lines. And so now that we have labels, we also need to show non-vessel things to the network. So we use the same kind of shapes that Malte used for registration. And now we have things that don't look anything like a real medical image, but has long and thin things that we want to segment, and then other types of shapes that we don't want to segment. It looks very naive, but then when we apply it to real data, it works pretty well. So this is one of those whole hemisphere MRIs. And what I'm showing in orange is a maximum intensity projection of the segmentation across a few slices. And we get anatomical things that we know exist in the human brain. So we have all these draining veins that go from the cortex to the sinus along the ventricles. And we've also worked on extending to OCT. So this is led by Etienne at the Martinos. And so the way we were making up vessels worked, but we needed to show artifacts that exist in that kind of data that didn't exist in the MR. So typically, the sort of striping that is caused by the acquisition process. So we just had to do the same, like don't have to be very realistic, but just needs to have features that we want to be invariant to. And this is what we get. On the left, this is a patch that has been manually segmented by someone very patient. On the right is what we get with the network. And actually the errors that we make are probably not errors, but just actually false errors made by the manual labelers that miss the very, very thin processes. So I'm quite happy with that. We're still working on validating these things. I don't have a very nice conclusion, but I'll just thank everyone who's involved in that project. Thanks, Steve.
Thanks, Yaël, for the great talk. Do we have any questions from the audience for Yaël?
So yeah, great talk. It's very nice to see all this, that you are able to recover this mesoscopic structure, vascular structure. Do you have any thoughts if this is easy to generalize to other kind of angiography imaging? For instance, to move this to clinical images for macro vessels, for instance?
So I tried to run the MRI network out of the box on time of flight data. It didn't work well, but I think it's because, I mean, first the range of radii that is done in time of flight is very different from what I was making up for the mesoscopic MRI. The other thing is that I think in the time of flight it would make sense to so we actually know like there is remaining signal from the parenchyma, right? We don't kill all of the signal from the non-vessel structures. So we have that in mind, but I haven't started training anything yet. But I think for angio, I would mix the SynSeg approach, where we use brain labels with synthetic vessels that we make run along the surface. But I think in that case, like knowing that you're on the PIA and not inside the brain is a pretty good prior to have. So it's definitely something I try to think about, but yeah, it doesn't work out of the box on angio.